So welcome everyone to everyone here in the auditorium and everyone joining us online. We're going to do the 2022 EMBL Alumni Award ceremony for the next hour and a bit. And it's my pleasure on behalf of the entire EMBL Alumni Board to host you, lead you through this program. My name is Erin Tranfield. I am based in Portugal, and I was here at EMBL Heidelberg doing my postdoc. So let's begin with the John Kendrew Award. Now this is a very special award. This award, I have the specs just to make sure I don't get it wrong. This award was founded in 2007 by Konrad Müller, but it has been very, very generously sponsored by Roland Specker since 2010, and I think Roland's joining us online, so can everyone give him a big wave as a hello and a thank you for the very generous sponsorship? Oh, and a round of applause too, thank you very much. I'm not actually going to take and do the big introduction, so with this very, very simple welcome, I'm going to turn it over to a PI here at EMBL Heidelberg, Detlev Arendt, and he's going to introduce this year's winner.
Thank you, Erin. My pleasure, my very special pleasure, to introduce Maria Tosches as our first John Kendrew Award winner. Maria actually is from southern Italy. She did an undergraduate in Pisa, then moved to EMBL starting 2007, 2008, and doing her pre-doc here and also then a first shorter postdoc. And then moved in 2014 to the MPI for brain research in Frankfurt, working with Gilles Laurent. And then in 2019, right good and in time for the pandemics, started her own lab at Columbia University in New York. And I chose, it might be slightly strange pictures to start with, but Maria, I chose it because it reminded me of our first lab retreat where you were there, which was on Sylt in 2010, and it was rather cold. If you look in detail, I think I'm the only one who is enjoying it, there in the middle. And everybody else looks a little hunched up. And then we learned from our southern Italian, so that 2 years later we were in Italy, on Procida. I think everybody is clearly more relaxed. If you look for the ones who really like lab pictures, there are 2 EMBL group leaders hidden there. So watch out. So this is Maria first and later. Then I was also choosing this title slide because it's actually stolen from a presentation I saw recently preparing for one of the excellence initiatives downtown, and it tells you that Maria is actually with her work also pioneered planetary biology research, because her work, her graduate work was chosen as a paradigm study for planetary biology. And this is what it was. It is her pre-doc work where she... Is the pointer actually working? Not.
I don't know, I didn't use it.
No? Should I try another one? Yeah, well, okay. In her thesis work in our lab, first she had to get accommodated to our worm. I mean, most of you will meanwhile know it, Platinares dumerili. It was also new to Maria. She came more from the vertebrate side and had to get accustomed with all the specialties of Platinares. And one of the specialties she then chose to investigate, which is that these worms have a larval phase and they travel upwards and downwards in a diel vertical migration. This is what it's called. So they travel upwards and downwards during the night. So say they have something like a day-night rhythm. And essentially what she found was that this is controlled by a melatonin sleep-wake cycle, if you want. So with a really impressive series of techniques that she had to establish for our worm, it's a mix of molecular biology that she did, for example, working at that time with zinc finger nucleases, of pharmacology, of establishing neurobiology techniques such as calcium imaging, and also a lot of imaging in general. She found out that there are cells in the worm here in red that release melatonin, so that is the hormone that also in us induces sleep at night. That this hormone controls cholinergic neurons in the worm that show rhythmic bursting and control ciliated cells that actually mediate the swimming upwards and downwards in the night. So that was a very intriguing result that what we thought is very typical for us sleeping and having the melatonin system in these worms controls the circadian rhythmicity and the diel vertical migration. So this was actually the main and big paper for her PhD. And what I want to emphasize is that Maria is not only a really meticulous experimentalist, she is also a deep thinker. And this is what I almost admired. She is going for the big picture. She wants to know what it all means, and she wants to really know. So when she publishes, she will always also publish something on top of that, which is a review. And here, for example, this review then deals with the significance of these results. Essentially, what she found in our worm is also found in the vertebrate hypothalamus, eye, and forebrain, and that these things seem to be evolutionary conserved and are referred to as apical nervous system. And I think this whole idea accumulates then in a new interpretation of the vertebrate brain that she did, in that the anterior part of the forebrain, called apical nervous system, is something that has fused with the sensory and motor alar and basal plates of the vertebrate brain into one forebrain, and this is referred to as a chimeric brain. So this was learning from the worm, about brain evolution in general. And but this is a vertebrate map, and you already realized that deeply in her heart, Maria has been interested in vertebrates always. So she, after her PhD and first postdoc, she moved into vertebrates, but not into a standard vertebrate, but she couldn't help herself and started with a focus on reptiles. She will tell us more about this in all the details, but I want to give you, so she also endeavored into single-cell sequencing, established for this organism single-cell RNA-seq, went deeply into describing cell types of the cortex in reptiles, and not only published a great paper, and she'll tell us about it, but also, again, thought deeply about the significance of these results. So what is old in the cortex and what is new? And how could something as intriguing as our cortex really evolve? And this was for her postdoc with Gilles, and then she couldn't help herself and established yet another vertebrate model system for her move to Colombia. This is now a newt, the Spanish newt or Plurodilis. She and her group are working on this now for 3 years, since 2019. She has already started. Again, you see the recurrent picture, a very nice data paper and a review, and explaining everything and going deep into the significance. So I won't tell too much about this. This, I think, is now on bioarchives and will hopefully come out soon. So Maria, I could not imagine a more deserving recipient of the John Kendrew Award. You deserve it for, I think, most of all for your exciting scientific vision on brain evolution. I think also for your powerful and really efficient experimental work, but what I want to stress is that it's always very careful and critical and also modest. I think you're never overshooting anywhere, so you with your interpretations. And basically for your fundamental insights into brain evolution. And if I have one message on top of this, I would really want you to keep your enthusiasm for evolution and for science in general. Yes, and I think the medals are both the same, right?
But if you mix them up, we'll solve it.
Also good.
That's the right one.
That's the one. Good. Okay, please, Maria. Congratulations, Maria.
Yeah. So good afternoon, everyone, to our live and to our virtual audience. Thank you, Detlev, for this nice introduction. And for resurrecting pictures from our lab retreats long ago. Still very fond memories of them. And especially thanks to the EMBL Alumni Association for selecting me as this year's awardee of the John Kendrew Award. This is a huge honor, and this is very special for me because my times at EMBL were really special times. This is where I came as a very naive, young, barely speaking English student, and I came out of here feeling that I could continue doing science, having that confidence, and feeling also as a citizen of the world. EMBL is such a magical place, and it's always a pleasure, and it's always exciting to come back here for conferences from time to time or to visit, and to see that this atmosphere, this vibe never changes, even though there's always new people and new research directions and new buildings. So I'm really excited to be here today, and I am proud of being an EMBL alumna. So what I would like to tell you about a little bit is my scientific path after EMBL, and where how the work that I've done here has planted the seeds for the rest of my career so far. So I've always been interested deeply inside in neuroscience. And understanding the brain, understanding how the brain functions, is one of the big frontiers in the big goals of science in general. And it's very hard because of the complexity of the nervous system. And big achievements have been made in the recent years and decades by taking an approach in neuroscience that I would define as the engineering approach, meaning looking at the brain as an assembly of neural circuits that process information from an input, take inputs, produce outputs, a little bit in analogy with computers. And this has been a really powerful way to study the nervous system and brains. However, it relies on an assumption, which is this idea that circuits are designed to be optimal and to be, that they are optimal to execute certain computations. But in reality, this is not how biology works. This is not how brains are produced, because brains have not been designed by engineers, but they are the product of an evolutionary process. And I think that we can complement that mainstream engineering perspective by thinking about the nervous system from an evolutionary perspective and trying to extract general principles of brain organization and function that make sense only in light of evolution. Because evolution is not a directional process, it does not have a goal, but it's just a progressive adaptation. It comes from the progressive adaptation of animals to their environments and thinkers with whatever is there. And many strange things about brain anatomy or brain function that do not make sense from an engineering perspective, they suddenly make a lot of sense when you think about them in a comparative perspective. So as Detlev mentioned, and I'm not going to spend too much time on this, from southern Italy, I started a journey towards the north when I first went to Pisa for my undergraduate studies. And there, as an undergraduate student, my first experience in the lab was by working on retina in frogs. And interestingly, retina and photoreceptors and amphibians have stayed with me in one way or the other, because photoreceptors were the cell types that I studied in platinum arrays here at EMBL. And when I came, I had no idea about evolution, to be really honest. The first lab meetings were really hard. People were talking about all these different animal phyla, invertebrates that I had almost not heard of. But the first question I asked Detlev, coming from that retina background, was why in the brain of a marine worm there are ciliary-type photoreceptors that we know are homologous to the rods and cones of our retina. What are they doing there? And so I think that question defined the main path of my work here at EMBL, which was an exploration of neurobiology in the marine environment. We took these platinum-arrays larvae, and we used them as a system to understand plankton neurobiology. these larvae have this girdle of cilia around their equator. They're using this cilia for swimming. And and as you can see in this video, ciliary beating propels the larva forward. Well, you cannot really see that in this video because the larva is immobilized, but the ciliary beating is modulated. Cilia change direction, sometimes they stop, they change beating frequency. And that modulation is achieved by nervous system control. You see here again the ciliated cells, multiciliated cells, and the nervous system stained in the middle. And in the middle of the larval brain, the ciliary photoreceptors that Detlev discovered here at EMBL, and that with molecular approaches I found, they also produce melatonin, the hormone that puts us asleep. And again, with the molecular approaches, I was able to see that melatonin receptor is expressed in these two cells labeled in blue down here, which are the motor neurons that directly innervate the ciliated cells. So establishing a very simple modulatory circuits where melatonin produced rhythmically according to the circadian cycle impinges on these motor neurons to regulate ciliary beating. And with experiments where we used pharmacology and electrophysiology and imaging, we could see that at night the melatonin signaling is increasing the activity of these motor neurons and increasing the frequency of ciliary arrest or ciliary stop. And I just want to show the experiment that I think was the most, and I remember very vividly, and it was the most exciting one for me, which is where I was able to image, using calcium imaging, genetically encoded calcium indicator, the activity of these motor neurons. You see the cell body here and the axon that projects, and you see these bumps of activity, which are happening at night when these cells receive the melatonin signal. So I still remember being at the imaging facility in front of the microscope in the middle of the night, and when I saw this, which we are, hypothesized would happen, but when I saw this in real life, it was one of the most exciting moments in my scientific life. And the lesson I learned from this work at EMBL more broadly is that you can use, now we are in exciting times where you can use modern genetic tools, imaging tools, to access and study nervous systems of any animal species, more or less. And so to really extend investigations on nervous system evolution, diversity and evolution. And this it's been something I've taken with me after EMBL. I was, as Detlev mentioned, I was deeply inside curious or more motivated to go back to understanding vertebrate brain evolution. And even though it's very hard to get to the point of human brain and minds, at least to start understanding the diversity of vertebrate nervous systems. And this is why I moved to Frankfurt, so still up north, working on turtles and lizards, and then with a jump to the west, to New York City 3 years ago, where we have started to work on salamanders. And before describing the science of this, I just also want to mention that Frankfurt was a time when I also started to get engaged with science communication, and we were going around in bars in Frankfurt and giving lectures on our work, of course, public lectures, and the Platinaries movies I showed you earlier were the favorite among the audience. So the biggest question in vertebrate brain evolution is the origin of the mammalian neocortex. It's the largest part of the cerebral cortex in mammals. It's the largest part by volume of the human brain, and it's called neocortex because it's new. There is nothing like that from an anatomical, ultrastructural perspective in any other vertebrate species, in any non-mammal. And the evolutionary origin of the neocortex has been a matter of debate for decades. And it's critical because the neocortex is associated to high cognitive functions, anything that goes from planning, decision-making to language. So to understand the evolutionary origin of neocortex, we have to look at the outgroup, or to the closest relatives of mammals. In our case, so in this case, reptiles and amphibians. And this is why I started the reptilian work in Gilles Laurent's lab in Frankfurt. And now in my lab, we're exploring the amphibian nervous system. If you look at the amphibian brain, if you look at the brains of these animals, even if you're not an expert, it is immediately obvious that by morphology, these brains have nothing or almost nothing to do with each other. Here we're looking at homologous regions of the brain, more specifically to the dorsal telencephalon, or pallium. And this is where the cortex is found, the neocortex in mammals, and these other territories that are called cortex in reptiles. And interestingly, in the same coordinates, the same position, there is dorsal telencephalon also in amphibians, but this has never been, by neuroanatomical criteria, never been considered or even called a cerebral cortex. And when we take a closer look, the defining feature of a cortex is layering. Layering is the alternation of cell bodies, of parts with high density of cell bodies, as you can see here, and parts with low density of cell bodies and axons and dendrites. And by that definition, the neocortex has 6 layers, the reptilian cortex has 3 layers, and the poor amphibians don't have any cortex in theory. This is how cell types or cell classes are distributed in these layers. So there are 2 main classes of neurons in the dorsal telencephalon of vertebrates. The glutamatergic neurons you see here in blue, they are the ones that congregate at high density to form the layers. And the GABAergic inhibitory interneurons that are scattered throughout the cortical tissue. So this is all we knew about cortex until a few years ago, and it was really unclear whether these neurons, for example, in layer 2 of the reptilian cortex, had any evolutionary relationship to any of the layers of the mammalian cortex. And just understanding or explaining how you go from a simple 3-layered cortex to a complex 6-layered cortex has been really a challenging question. Thankfully, or at least luckily for us, we've been able to use new molecular approaches, single-cell RNA sequencing, to start to dissect the molecular identities of these neuron types and classes, and to compare them in a quantitative way, systematically, across species. And to essentially take this problem that has always been in the realm of comparative neuroanatomy into a new field, the comparative single-cell RNA sequencing. And here you have just an overview of the samples and the data sets that I've collected while in Frankfurt, studying a turtle and a lizard, the bearded dragon lizard here. And this is the data from my new lab at Columbia. We can isolate single cells, sequence their transcriptomes, use sophisticated bioinformatics approaches, which I don't have the time to talk about, to compare these transcriptomes. And from all this work, what we learned is that glutamatergic and GABAergic neurons have followed different evolutionary paths or different destinies in the vertebrate history. So let's start from GABAergic interneurons. And this was evident already from the raw data. What you're seeing here is a matrix where for each gene, marker gene in rows, there is expression in each single cell, which is in columns. And this is from mouse data, publicly available. And this shows that from these marker genes that have been thoroughly characterized in the literature, you can already identify 3 major clusters that correspond to the 3 classes of GABAergic interneurons in the mammalian neocortex. So the first thing I did was to take this list of genes that are so very well known and characterized and ask where are they expressed in, or how are they expressed in GABAergic interneurons in a turtle, for example. And what was very stunning to see was that these genes alone are already sufficient to group turtle GABAergic interneurons in 3 classes, which are the same 3 classes that have been identified in mammals. And this extends also to salamanders. So that means that the GABAergic interneurons types, and now we know also some of the subtypes that have been described in mammals, are not mammalian specific, but they are instead present already, they were already present in the ancestor of reptiles, amphibians, and mammals at the very least. This is important because GABAergic interneurons are extremely specialized. They have specialized functions in cortical circuits, and so that suggests that perhaps some of these functions are very ancient in vertebrate nervous systems. And the picture is completely different for the other neuronal class, the pyramidal neurons. These are neurons that are assembled in layers, in the 6 layers of the mammalian neocortex. And again, just looking at the raw data, you can see that there are genes. Many of them are transcription factors that enable us to classify and to distinguish between these different pyramidal types, layer 2, 3, layer 4, layer 5, and so on. And if you take the same genes and you look at them in turtle pyramidal neurons, you can see that they are either expressed everywhere or they're expressed nowhere, meaning that the molecular logic, the molecular identity of glutamatergic neurons in reptiles and also in amphibians, we know now, is completely different or dramatically different from the molecular identities of pyramidal neurons in mammals. And this is summed up here, where I'm trying to show with different shades of blue the different species-specific identities of these pyramidal types. However, we also see that there are certain groups of genes, so aspects of the molecular signatures of these neurons that are related to the order of birth during development. So we found, surprisingly, that also in reptiles and in amphibians, these pyramidal neurons are not all the same, but there's different types that are stratified in layers that cannot be recognized by anatomy, but can be instead recognized by molecular markers. And these layers are produced sequentially during development, exactly like in the mammalian neocortex. They take up a different location because in mammals there's been another innovation, which is the migration of neurons on top of their predecessors. So the late-born neurons migrate on top of the early-born neurons. There's been an inversion of the gradient of corticogenesis. But nevertheless, there are some genetic signatures that are shared between early-born neurons across all these tetrapods and late-born neurons across these tetrapods, suggesting that some of these developmental constraints are still shaping and determining the organization of the cerebral cortex in all these animals. So to summarize, the work I've done so far has been reminding me that brains are highly specialized, are flexible, and are evolvable. And there are these species-specific characteristics that can be extracted from comparing molecular data and other types of data, obviously, and that remind us that by comparing brains, we're looking at these outcomes of evolution that are amazingly fit somewhat to their environment. And this also, the hope of, the goal of our research is to extract general principles of brain organization and function that are invariant of the species-specific characteristics. But I think, more prospectively, another hope is that by learning about the past of brains, we may prepare in some way also to the future. Brains are evolvable, change, and will continue to change. And for example, things like the new development in machine learning and computer-machine human interfaces are certainly going to shape the way our brains develop and interact with the environment, as well as the changes in the environment and climate change, which are expected to have an impact on brains, especially in cold-blooded animals. So I'm hoping that by extending comparative work, we can at least, or the field can infer some of the principles that may help anticipate some of these or understand some of these changes. And with this, I would like to conclude by thanking Detlev and members of the Arendt Lab, past from with whom I was lucky to work, and thesis advisory committee and other mentors, friends, and colleagues at EMBL. My postdoctoral mentor, Gilles Laurent, and people in the Laurent Lab, and especially the people with whom I worked more closely that are listed here. And my colleagues and collaborators at Columbia and other institutions, and friends and family. My lab, with these brave people that in the middle of a pandemic came to work on an animal that none of us had ever worked on before, and our funding sources. And thank you again for this award, and thank you for your attention.
Thank you very much, Maria. That was a really fantastic talk. What an inspiring story, and what a great beginning to this ceremony. I tootled through it pretty quickly, but I'd like to emphasize that all of the information about these awards is available on the EMBL Alumni site. And if you're interested in knowing more about any of these awards, please do look there. There's also a beautiful photo gallery, soon with a new picture added to it, of all the current and past recipients of this award.